CBC Part 2, Hemoglobin, HB. Welcome to this video on hemoglobin, one of the most important parameters measured in the complete blood count, or CBC. Hemoglobin, commonly abbreviated as HB, is the oxygen-carrying protein found inside red blood cells. Its primary function is to transport oxygen from the lungs to body tissues and carry a portion of carbon dioxide back to the lungs for exhalation. Measuring hemoglobin is an essential part of the CBC and helps healthcare professionals diagnose anemia. Structure of hemoglobin. Hemoglobin is a complex protein composed of two main components, globin and heme. The globin portion consists of 4 protein chains, 2 alpha chains and 2 beta chains in normal adult hemoglobin. Attached to each globin chain is a heme group, which contains a central iron atom. Each iron atom can bind 1 oxygen molecule, allowing a single hemoglobin molecule to carry up to 4 oxygen molecules at the same time. This remarkable structure makes hemoglobin highly efficient at transporting oxygen throughout the body. When we inhale, oxygen enters tiny air sacs in the lungs called alveoli. These alveoli are surrounded by microscopic blood vessels known as pulmonary capillaries. As red blood cells pass through these capillaries, oxygen diffuses into the blood and binds to the iron atoms in hemoglobin. This forms oxyhemoglobin, which carries oxygen through the bloodstream to every part of the body. As oxygen-rich blood reaches the tissues, hemoglobin releases oxygen where it is needed. The oxygen enters cells and is used to produce the energy required for normal body functions. After releasing oxygen, hemoglobin also binds a portion of the carbon dioxide produced by cells and transports it back to the lungs for exhalation. Normal hemoglobin values. Normal hemoglobin levels vary according to age, sex, and laboratory reference ranges. Typical adult values are males, approximately 13 to 18 grams per deciliter. Females, approximately 12 to 16 grams per deciliter. Causes of low hemoglobin. A low hemoglobin level is known as anemia. Common causes include iron deficiency, vitamin B12 deficiency, folate deficiency, blood loss, chronic kidney disease, bone marrow disorders, hemolytic anemia. How hemoglobin is measured in the laboratory. In modern laboratories, hemoglobin is measured automatically using a hematology analyzer. First, a special reagent is added to lyse or break open the red blood cells, releasing hemoglobin into the solution. The analyzer then measures the hemoglobin concentration using spectrophotometry. In this technique, a beam of light passes through the hemoglobin solution, and the instrument measures how much light is absorbed. The amount of light absorbed is directly proportional to the hemoglobin concentration, allowing an accurate measurement. The result is then reported as part of the complete blood count. Hemoglobin can also be measured manually using Sally's hemoglobin meter, also known as the acid hemeton method. In this method, 0.1 normal hydrochloric acid is placed in the graduated Sally tube. A measured volume of blood is then added, converting hemoglobin into acid hemeton, a brown-colored compound. Distilled water is added drop by drop while mixing until the color of the solution matches the standard comparator. The hemoglobin concentration is then read directly from the graduated scale on the Sally tube and reported in grams per deciliter. Although Sally's method is simple, inexpensive, and still used in some resource-limited settings, automated hematology analyzers provide much greater accuracy, precision, and reliability. Thank you for watching. If you found this video helpful, please like, subscribe, and stay tuned for more medical laboratory science videos.